On behalf of my co-authors and partners at Verana Health and the American Academy of Ophthalmology, thank you for allowing me to present our work on replicating the clinical trial outcome data from the View 1 and View 2 studies. Here are our disclosures. Relevant here is that I am a consultant to Verana Health, and my co-authors are either employed by Verana Health or the American Academy of Ophthalmology. Neovascular age-related macular degeneration is the leading cause of vision loss in adults over 60, and the View 1 and View 2 studies were phase 3 randomized clinical trials that led to the FDA clearance of aflibercept in 2011. The View studies assessed the non-inferiority with a margin of 10% of the aflibercept arms to maintain vision at 52 weeks when compared to monthly ranibizumab. Maintenance of vision was defined as a loss of less than 15 ETDRS letters. Here, we describe the results of an analysis of the American Academy of Ophthalmology's IRIS Registry and compare the real-world outcomes in neovascular AMD patients to those reported by the View studies. The IRIS Registry was the first comprehensive eye disease clinical dataset based on electronic health records. Over 70% of US ophthalmologists have contributed 367 million patient visits from 66 million unique patients to the database, which contains 7 years of longitudinal data. In order to replicate the View trials as closely as possible, we adopted the same inclusion and exclusion criteria. We included patients older than 50 with treatment-naive neovascular AMD with a baseline vision between 20/40 and 23/20. If 2 eyes of the same patient met study criteria, we used the first eye to have neovascular AMD as the study eye. We excluded any patients with a prior diagnosis of neovascular AMD, diabetic retinopathy, diabetic macular edema, uncontrolled glaucoma, corneal neovascularization from causes other than AMD, prior retinal detachment, vitreous hemorrhage, macular hole, uveitis, vitrectomy, or corneal transplantation. Eyes were also excluded if they previously had ocular or systemic AMD treatments and if gender or race information was missing. We were able to replicate 3 arms from the View studies. Arm 1 received aflibercept every 4 weeks with a maximum of 12 injections. Arm 2 received aflibercept every 8 weeks after 3 monthly loading doses with a maximum of 8 injections. And Arm 3 received ranibizumab every 4 weeks with a maximum of 12 injections. There were 1,632 subjects enrolled in the original View studies, meeting these criteria that were ultimately analyzed in what was specified as the per protocol set, or PPS. Next, I will describe the process we used to create an IRIS registry cohort to match this. As this is a retrospective study using real-world data, we first had to determine if there was data available in the IRIS registry that would allow us to build a comparable cohort. Fortunately, all 3 of these View inclusion criteria were able to be addressed in IRIS. When it came to the View exclusion criteria, we were able to apply them partially with the data available in the IRIS registry. Eyes were able to be excluded with prior AMD treatments, anti-VEGF treatments, vitreous hemorrhage, CNV, vitrectomy, retinal detachment, macular hole, surgery within 3 months of the index date, and any previous diagnosis of diabetic retinopathy, diabetic macular edema, or retinal vein occlusion. There was insufficient data in the IRIS registry to exclude eyes that may have been treated with an investigational agent for neovascular AMD. Moreover, as it was not present in the registry, we could not analyze fluorescein angiograms to determine lesion size as was performed in the View trials. Similarly, there were no color fundus photos available to determine if there was subretinal hemorrhage, scar, fibrosis, atrophy, or RPE tears that would have excluded an eye from analysis. Given the available data, we determined the file cohort through an attrition process, applying IE criteria at each step. In step 1, we initially found 605,482 eyes that received either aflibercept or ranibizumab with known laterality between January 1st, 2013 and December 31st, 2018. After applying that criteria, we were left with 99,093 patients for our IRIS registry cohort. Unless there were time constraints specified, we looked back to the earliest entry in the database to apply all the exclusion criteria. To summarize, the eligibility criteria for inclusion in the study were patients older than 50 years with a neovascular AMD diagnosis that were treatment naive on the index date, and who met the VIEW study inclusion-exclusion criteria available in the IRIS registry. We evaluated patients that met 3 treatment strategies used in the VIEW studies. That included Arm 1, that received aflibercept every 4 weeks, Arm 2, that received aflibercept every 8 weeks after 3 monthly injections, and Arm 3, that received randomizumab every 4 weeks. The follow-up period began at the treatment start date and ended 1 year later. The outcome recorded was the best corrected visual acuity in the IRIS registry at 365 days, allowing for plus or minus 2 weeks. Similar to what was done in the VIEW studies, patients missing visual acuity measurements at 1 year from their treatment start date had their 1 year visual acuity measurement imputed from their last post-index visual acuity measurement. Eyes were analyzed using the VIEW study's per protocol effect, meaning that in our study, the monthly treatment groups could have missed up to 2 doses, and the 2Q8 group needed to receive 3 monthly loading doses followed by an injection every 8 weeks with no more than 8 doses and no missed doses. As in the VIEW studies, our primary outcome measure was the proportion of patients who maintained vision, meaning they lost less than 15 letters at 52 weeks. The secondary outcome measure were mean change in vision and the proportion of eyes gaining 15 or more letters. Once we had our 99,000 patients, we looked at the treatment patterns of those eyes with the goal of replicating the treatment arms of the VIEW studies. Ultimately, we were able to include 4,779 patients in our per protocol set, with 2,730 eyes that receive aflibercept every 4 weeks, 165 eyes that receive aflibercept every 8 weeks after 3 monthly loading doses, and 1,884 eyes that receive ranibizumab every 4 weeks. Here are the baseline characteristics of the original VIEW studies. Note that the mean age was 73 to 78 years, and the mean baseline vision was 51 to 55 letters. Also, the VIEW participants were about 56 to 63% female and 71 to 97% white. When looking at the baseline characteristics of the IRIS registry study arms, we found them to be comparable in most measures. However, our cohort was older at about 79 to 80 years, and the baseline vision was also better at 57 to 59 letters, which is about 1 line better vision than the VIEW studies. Also, there were more females in the IRIS registry cohort at 59 to 64%, and there were fewer white patients than VIEW 1, but more than in VIEW 2. Now, let's take a look at the real-world outcomes of our 3 arms. For the primary outcome measure, data from our IRIS registry analysis showed that the proportion of eyes maintaining vision was similar to the outcomes of the VIEW studies, with 90 to 94% of eyes losing less than 15 letters. While there was a slight advantage to the 2Q8 arm, a Wilcoxon test on the imputed logMAR difference values were all non-significant across all pairwise treatment groups at 1 year. The proportion of eyes gaining 15 or more letters were very similar across our study arms and ranged from 19 in the 2Q4 arm to 23% in the RQ4 arm. While similar and non-inferior across the board, the real-world outcomes were lower than the visual acuity gains in the VIEW studies. Perhaps this difference could be attributed to the fact that the VIEW participants had worse baseline visual acuity when compared to the IRIS arms, and thus were less sensitive to a potential ceiling effect than the IRIS patients. When we looked at the mean change in vision, the 2Q4 arm improved by about 3 letters, while the 2Q8 and the RQ4 arms improved by 5 and 4 letters, respectively. Again, there was less overall mean improvement in the IRIS registry study when compared to the VIEW trials, which may be influenced by the better baseline vision of the IRIS arms. Finally, here's an analysis of the risk difference of maintaining vision. On the left side, you have the results of the VIEW studies, and on the right, our IRIS registry study. If we use the original VIEW studies inferiority margin of 10%, what both studies show is that the 2Q8 aflibercept arm was non-inferior to the RQ4 arm. In summary, The IRIS registry arms had similar outcomes when compared to the phase 3 VIEW 1 and 2 studies, indicating a proof of concept and demonstrating that, to my knowledge, we could for the first time use real-world data to replicate the primary outcomes of a large-scale clinical trial in ophthalmology. Benchmarking real-world endpoints versus randomized controlled trials could be a valuable tool in validating the predictability of these endpoints as a measure of treatment effectiveness. A full description of our analysis is forthcoming in a manuscript. Thank you very much.